My name is Holly May-Lewis. I'm a PhD student at the University of Surrey, supervised by Professor Roger Webb and Professor Melanie Bailey. I have been optimising a spatially resolved extraction technique called direct analyte probe nanoextraction, also known as DAPNe, which I have combined with liquid chromatography to analyse discrete areas of a sample. This presentation discusses the use of DAPNe-LC-MS to detect drugs and lipids at a single cell level. Obtaining a detailed molecular analysis from a single cell is important in many fields of biomedicine. Although neighbouring cells share similar genomic information, the cells are heterogeneous due to different metabolic pathways and epigenetic differences. Sensitive, reliable and high spatially resolved methods need to be developed to probe cells. Direct analyte probe nanoextraction, also known as DAPNe, is a technique that uses a nanocapillary driven by a nanomanipulator and is combined with mass spectrometry. Due to the small tip diameter, DAPNe has the ability to extract a single cell. A cell of interest is selected using a microscope. DAPNe is then used to extract the single cell into a tip, and a mass spectrum of the contents of that tip are required. The cells used in this study were Pink 1 cells and were cultured by Priyanka Gupta at the University of Surrey. The cells were spiked with 100 micromolar of 5 different anti-tuberculosis drugs and were incubated for 24 hours. Before DAPNe analysis, the cells were washed 3 times with PBS, and 5 ml of the PBS solution was added to the petri dish. The cell of interest is located using a microscope. The capillary tip of chosen tip diameter, in this case 15 micron, is inserted into a nanomanipulator. The tip is lowered in small increments until just above the cell. A back pressure is applied to pull the cell into the capillary tip. A solvent can then be added to the back of the tip to dissolve the cell, in this case 50-50 methyl or ethanol. The contents of the tip were then pushed into an LC bowl and analysed using liquid chromatography mass spectrometry. In total, 30 cells were extracted and analysed using DAPNe. The only anti-TB drugs that were detected were ethambutol and dapnone. In 2 out of 30 cells, no drugs were detected, but in the other 28, both these drugs were detected. In the successful extractions, the drug peak intensity was normalised to the cell volume, which was approximated using the microscope software. It can be seen that there was a high variation in drug intensity, which could suggest cell-to-cell drug uptake heterogeneity. The LC-MS method used also successfully detected lipids in the single cells. A software called lipid search was used to identify the detected lipids. The total lipid intensity for each cell is shown. For cell 10 and cell 20, where no ethambutol or dapnone were detected, the lowest lipid intensity was seen. Although the number and intensity of lipid features varied, the lipid profiles did not vary considerably. Phospholipids were detected at the greatest frequency, which is most probably due to the phospholipids seen in the cell membrane. To conclude, DAPNe can successfully be used to extract single cells. DAPNe-LC-MS can successfully detect drugs and lipids simultaneously within a single cell. It was found that there was cell heterogeneity between the cells in the same culture. Whilst the number and intensity of lipid features per cell vary considerably, the lipid profiles from cells where a high frequency of lipids were detected was relatively consistent. Thank you for listening to my presentation. If you have any further questions, please do not hesitate to contact me. Thank you.